All right, it's a new unit in biology. This is Unit 3, the structure and function of DNA and RNA. All right, so we just finished the processes that happen in cells with mitosis and meiosis and how that lets you grow and reproduce on a large scale. We're going to start talking a little bit about how that actually happens, though. So all our genetic information is stored in DNA, and we're going to look at the relationship between those today. All right. We will get to how new genetic information is formed, and we're talking about new here. We're talking about the same information, but new DNA. So we've got one strand of information, DNA, and we're going to create a copy of it. So same information, new DNA structure. So before we get into all that, we got to do a little bit of a history lesson. And I'm not one that's great on the numbers and everything, but the people and what they did is the important part here. So Friedrich Miescher is the first person to actually isolate the DNA molecule. He called it a nucleon. 12 years later, in 1881, Albrecht Kossel named this nucleon deoxyribonucleic acid, or DNA. He also named the 4 nitrogenous bases, which we'll cover a little bit later in the video. So remember that deoxyribonucleic acid is DNA. DNA is deoxyribonucleic acid. So we've got a D, an N, and an A. All right? It's just a initialism or an acronym. In 1941, I lost a 1. In 1944 to 1950, Erwin Chargaff worked on equating Mendel's research with traits and their inheritance with this new DNA and the 4 nitrogenous bases, and he posited and published that DNA is how traits are inherited. In 1950s, here in America and overseas in Britain, there was a little bit of drama in working towards the structure of DNA. Right? So in 1951, Rosalind Franklin took this picture. It's called Photo 51 in her writings that shows this X shape. And she spent a year studying this to figure out that DNA is shaped like this, a double helix. During that year, however, someone else took this photo to Watson and Crick and showed it to them. They had also been working on finding out the structure of DNA, but they'd been approaching it from a different angle. They used this photo to narrow down their own theories into an actual published work that said DNA had a helical structure. They ended up publishing at the same time that Rosalind Franklin did, and because of the views on women scientists at the time, which were unfortunately incorrect, they put Rosalind's paper at the end. So Watson and Crick's paper happened first before Franklin's paper. All right, so we had one, if you were reading it like a book, one would show up before the other. And this led a lot of people to believe that Rosalind Franklin had just done work to support Watson and Crick's publication, when in actuality, they were doing it very independently, and Watson and Crick had actually used some of Rosalind Franklin's work without her knowledge. I'm going to have my bio class watch a video on this, so I'm not going to go too much in depth here, but I think it's important to know that science research does come down to who publishes first and who can prove that they did it. Rosalind Franklin could prove that she did it, and she published, but she published pretty much at the exact same time. And because later Nobel Prize being awarded for this only works towards people who are alive, and Rosalind Franklin died of cancer, Watson and Crick got the Nobel Prize, and they got a lot of press for it. That's unfortunate. But nevertheless, we will learn from the mistakes of the past. All right, speaking of past, Nucleic acids. You should already know that nucleic acids are a complex macromolecule made of repeating nucleotides. This whole slide is straight out of last unit. I copy and pasted it. It's mine. I can do that. Right? So we've got a phosphate group, a sugar group, and a nitrogenous base. Remember that the nitrogenous bases are nitrogenous because they have nitrogen. That's these little N's. All right? Whereas a sugar looks like a sugar. And phosphate groups, it's got a phosphorus and 4 oxygens, which is phosphate. If you take these and repeat them in a structure, they're going to start looking like this, where those phosphate groups are actually going to bond to the sugar groups, and they will form one half of this, like the left side or the right side. Now, because those nitrogenous bases fit perfectly with some of the other nitrogenous bases, we get something called base pairing or base pairs. So the 4 nitrogenous bases, I told you we'd get to them, are adenine, thymine, guanine, and cytosine. All right, and they are grouped as such. Cytosine can bond to guanine. Guanine can bond to cytosine. All right, so they will pair up. They will link. Adenine and thymine work the same way, okay? But they won't work if you take a cytosine and try to get it lined up with a thymine. It just doesn't fit, right? Chemically, they will not bond because they are the wrong shapes. That's the easiest way I can explain it and the most correct way without oversimplifying it and causing some errors in thought. Right? If we take that model and we zoom out on it a little bit, we're going to get something that looks like a ladder, where the glucose, still this blue color, right, forms the outsides of a ladder where the nitrogenous bases form the individual rungs, the things you stand on. You zoom out a little farther, and we get this double helix shape over here. All right, and it starts to twist due to something called hydrogen bonding, where hydrogen is going to be pulled towards a negatively charged portion of another molecule or of the same molecule, depending on how big it is. And if we zoom out farther, we're going to get a coil and then a supercoil and a chromosome. All right, so if you've ever taken a shoestring or a piece of yarn and twisted it, you'll start to see that it coils, and then those coils will coil on themselves. They'll like spin around themselves. This is what DNA does. So DNA will start to wrap around something called a histone. You don't need to know what it is. I'm just talking about it. All right. Those histones, and as they kind of coil around them, are called nucleosomes. Again, this isn't super important for you to know. It's just something we're talking about. Okay. So those nucleosomes are going to twist together and tighten up and get closer and closer together until they can't anymore. Then they're going to start to spin around one another, forming these coils. All right. I really like this picture because you can see that they start to do that. All right. And as those coils try to get closer and closer to one another and spin closer and closer to one another, we get something called a supercoil. Right? Supercoils will also coil around themselves and fold and bend into chromosomes. All right, we've talked about chromosomes a little already when we talked about mitosis and meiosis. So just linking it to what we already know. Right? That's it for this video because I also want my own class to watch a video on Rosalind Franklin and Watson and Crick and the stuff that went down there. Next time we will talk about how new genetic information is formed, i.e. copying. Right? If you've got questions and you're one of my students, send me an email, reach out, post on the questions and reflections forum, and hope everybody has a good day.